yesterday, since we were talking about Campylobacter. There's an outbreak that just happened in Pennsylvania that was linked back to some raw milk. So it does appear there have been some cases of campylobacteriosis associated with this outbreak. Sometimes happens, doesn't, and sometimes doesn't happen. With that cucumber salmonella outbreak I was mentioning the other day, there were no cases associated with it. They just identified it during routine surveillance, I guess, and they pulled 71 boxes of cucumbers and destroyed them. But with this, unfortunately, they found out because people were starting to get sick and displaying some of these foodborne symptoms associated with Campylobacter. So, all right, gram-negative bacteria. We have this kind of a hodgepodge deck. We have some cocci that we're going to be talking about, which we will hopefully get into today. A lot of these are also gram-negative rods. Some really important species. A lot of these are less common. So we do have some anecdotes and some vignettes and some examples of outbreaks to kind of illustrate the impact of some of these. And some of these are also biological weapons, or they can be weaponized. So we'll be talking about those as well. Most of these are facultative anaerobes. There are a couple of exceptions, but most of them tend to do very well or thrive in oxygenated sites. We're still talking about gram-negative, so remember that we have LPS and then we have endotoxin release. And as a result, some of these symptoms are associated with inflammation and immunopathology as well. These major pathogens, the family of Pseudomonads includes both Pseudomonas and Burkholderia. They have a huge array of different diseases and infections that they can cause. But as is the case with many of these two genera in particular are usually going to be opportunistic or cause opportunistic infections. We have a bunch of different genera that are associated with respiratory tract infections. I guess you could also include at least one species of Burkholderia onto that list too, Burkholderia cepacia. It's important, but we're not talking about that one specifically. But it causes respiratory infections. In terms of morphology, cocci and cocobacilli, that's CB nomenclature. Do you remember from block 1 when we were talking about morphology and we were talking about either rods or spheres, bacilli or cocci? There is that hybrid, and that's what we're talking about here. They're really short, stubby, kind of plump-looking rods or cocci, depending on how you view them. We also have a bunch of different zoonotic organisms. We've briefly talked about what zoonotic organisms are. They are infections that are usually maintained in animal reservoirs that we can catch from those animal reservoirs. Salmonella, some of those species, Typhimurium enteritidis, for example, some of those Campylobacter species would potentially be considered zoonosis, but we get it through ingestion. And some of these, they're more direct contact with animals, so we'll talk about that a little bit more. Like Burkholderia, for example, classic example, Burkholderia mallei, the zoonotic disease you can get. And then lastly, spirochetes. We won't get to that until Monday. That's towards the end of the deck. But that was a morphology that we talked about with those very like spiral corkscrew type bacterial cells that can cause some very serious and significant diseases. So we'll start off with Pseudomonads, which is a very, very diverse group of bacteria. Their morphology is usually pretty rod-shaped, at least when you're looking even under oil, like 100x oil of immersion. You won't be able to see that curvature, but they are usually slightly curved like this. And for motility, this one is showing quite a few flagella at one end, one polar end. So they can be multiflagellated, locotrichous, a bunch of different arrangements. It just depends on the particular species of that. There is a lot of movement within these different taxa, so taxonomists are recategorizing some of them, and that is sometimes difficult to keep up with, but I'll try to keep it as simple as possible. In terms of respiration, most of these are obligate aerobes. Some of them can use nitrate, NO3, as a terminal electron acceptor in their electron transport chain. So they have a little bit of expanded conditions, and they can undergo anaerobic respiration as well. And again, these are the two genera that are included in this family. When we're talking about the genus Pseudomonas, There are some other clinically relevant ones, but Pseudomonas aeruginosa is by far the most clinically relevant member of this particular genus. It's not a strict human pathogen. It's found out in the environment, on plants, and in the soil, and in water. And even in humans, because it's a fairly robust organism, it can transiently and infrequently colonize some of these areas like our skin and our respiratory tract and some of those other areas. But again, those are usually transient colonizations that occur. Most of the infections that we worry about happen in hospital settings. And the 50,000 infections, hospital-acquired infections that happen annually just in the US are testament to the fact that we have a very difficult problem eradicating these bacteria from those settings. Most of the deaths that occur every year are among the most critically ill people who are susceptible to these infections. And also, I'll come back to this point, but the antibiotic resistance doesn't help with that fact. We have now multi-drug resistant strains of Pseudomonas aeruginosa, extensively drug-resistant strains, XDR, PA, and then we have pan-drug resistant strains as well, which is really, really kind of a horrifying thing, resistant to all classes of antibiotics. In terms of their versatility, because they are environmental bugs and they can survive in all these different environments, one of the other features that allows them to take up residence in hospitals and prevent being destroyed when hospitals are trying to clean and disinfect is that they tolerate a very wide range of temperatures, so they can grow in places like hot water baths, in hot tubs, in laboratory water baths. They can do fine in IV tubing. They're associated with ventilators and can cause ventilator-associated pneumonia bath. So they do very well. They're very rugged. In this picture, you can see another virulence factor that we talk about all the time, but you can see it on the gram stain. What feature are we looking at in that gram stain of these gram-negative rods? Capsule. Yeah, you can very easily see this is a capsulated strain. This one, I don't know if it doesn't have it or it's just not expressing it, but this is the absence of a capsule, just the pure gram-negative rods. And then on a plate, some of these strains will produce molluscins, so you'll see some clearing. And this is a strain that is hypermucoid, kind of what we saw with Klebsiella pneumoniae, how mucoid that was. Super mucoid looking colonies because they produce that capsule. In terms of pathogenesis, we have our normal attachment factors, the Fimbria and Pilla, that are responsible for attachment to whatever tissue, and these literally do attach to a wide range of tissues in the body. So they have that going for them. But more importantly is their alginate capsule, which is sort of like, it's like a biofilm for an individual cell. It's a very thick, gooey, slimy layer. It's just like when you see rocks that are covered with an alginate cover, how slippery they are. It's like that on these individual cells. So this electron micrograph on the right shows these naked cells without much alginate capsule covering them, but they can express that, they can induce it upon getting some environmental signals to do so, and they start expressing this heavy capsule, this alginate capsule. And then when they get a thick layer of alginate going to protect them, and they start accumulating many cells, many layers, that will provide protection against antibiotics that we've talked about before and some of these other things. They also produce a ton of different toxins, a lot of different enzymes that will help promote invasion and cause some of these opportunistic infections that we'll talk about. With these toxins, they produce a toxin called exotoxin A, which is toxic to cells that are intoxicated with it because it stops protein synthesis. So if the cells are producing, this bacteria are producing exotoxin A, they deliver it not directly with a type 3 secretion system, but with a type 2 secretion system. So they secrete it outside of their cell into the environment, and that exotoxin A will go out and enter some of these target cells and basically kill them by blocking protein synthesis. So that's responsible for some of the tissue damage that we see with some of these opportunistic infections. We also have pigments that are produced by this bacteria. In this preceding slide, we didn't see a lot of pigment here on these colonies, but I don't know if I have a picture later that shows it. They can produce some very bright pigments. Here, we're talking about pyocyanin, which is blue colored pigment. There's pyoverdine, which I misspelled, it has an E on the end of it. There's also pyomelanin, which has a brownish color to it, and pyrorubin. So all of those are not just pretty colors, they actually do things to contribute to the virulence of Pseudomonas aeruginosa. Pyocyanin, for example, is useful because it catalyzes this reactant oxygen species, which can cause tissue damage. It's like hydrogen peroxide and superoxide. Pyoverdine is a siderophore, so it'll sequester iron from the host. That can cause some damage as well. And then in terms of these enzymes that can be produced, these invasive enzymes, all of these, elastases, proteases, including some immunoglobulin proteases that'll destroy IgA and IgG, these lipases, phospholipases C, all of that contributes to the tissue destruction that you see, particularly with burn patients and patients with wound infections. Oh yeah, I'm sorry, so here's a plate up here. Just a normal plate, and you're growing out Pseudomonas aeruginosa that has this pigment that's being expressed. And it's very, light green color here. Some of this is fluorescent, so particularly that green pigment, if you put it under UV light, it'll fluoresce. Other species of Pseudomonas can do that as well. But you can even see that green pigment sometimes in like a burn wound, you can see here, or in nail infection, or here where somebody has keratitis, an eye infection being expressed there. There are other bacteria that can cause that pigmentation, so if you see a green infection, it only should raise the index of suspicion for Pseudomonas aeruginosa, but you still need to get a lab result or confirmation of that. In terms of types of infections, there's some localized infections we'll start off with, and then there's some systemic infections. For localized infections, we have a couple different types of ear infections. The first is just routine swimmer's ear, externa otitis, where you have that moist ear canal that gets an infection, and it's usually self-resolving. It's very common, not a huge problem. And then we have something else entirely when we talk about NOE, necrotizing otitis externa. This is usually associated, so normal swimmer's ear, that's anybody, any healthy person who has this dampness in their ear and they get exposure to this bacteria. But when you're talking about this very serious, potentially life-threatening form, necrotizing otitis externa, this is usually in people who are immunocompromised or super old, and it is aggressive and can cause osteomyelitis. It destroys the temporal bone next to the ear there. And you can get meningitis as well as it starts spreading inside the interior. That's just because of the extent of the invasiveness of the bacteria, those invasive enzymes. So eye infections, these are oftentimes associated with contact lens wearers who are washing off their contacts in non-sterile solutions or water that has Pseudomonas aeruginosa in it. So those can be very, very serious, like this type of infection. Skin infections, they range all from fairly mild, like hot tub folliculitis, where you just get a little bit of, like this guy here. It's not super high resolution, but you can see this is just bacteria that have probably gotten into hair follicles and caused like this rash. Fairly mild, self-resolving generally, all the way up to things like infections in people who have burns over a significant portion of their body. And that's very, very difficult to treat, very difficult to clear. Again, because of that broad temperature range from 4 degrees to 42 degrees centigrade, they do well in some of these hot or warmer temperatures. Outbreaks have been linked to people who get pedicures at spas and they're dipping their feet into those tanks, and they get Pseudomonas aeruginosa infections because they have a little break in their skin and bacteria can get in that way. So that's something to kind of link with that. And then burns, this is an important point. If you survive the burn itself, the main cause of death is Pseudomonas aeruginosa infections. So UTIs and hospital-acquired settings are common, as are a lot of these. And broad-spectrum antibiotics can be a predisposing factor for development of UTIs, Pseudomonas aeruginosa. A few more localized infections to have on your radar are some respiratory infections. VAPS, again, ventilator-associated pneumonia, where you already have patients who are struggling, critically ill, and now you're putting them on a ventilator that has contaminated water, so it's mainlining those bacteria directly into their lungs. So that's very, very dangerous. And then we also have this association with cystic fibrosis. I highlighted this on your screen because it's so important to be aware of this connection between Pseudomonas aeruginosa and cystic fibrosis. Anybody know what cystic fibrosis is? Yeah. Yeah. So yeah, it's a genetic disease where you're born with this disease, and you cannot basically get the consistency of the mucus in your lungs to the right viscosity. So it becomes very thick, and it's not able to function the way it should, which, as we know, in the airways, that mucus needs to be able to trap bacteria and then lift it up out through the mucociliary elevator. So if you can't do that, basically you become an incubator for a bunch of bacteria to grow, and it becomes very, very dangerous. Life expectancy for people with CF has historically been very low, like people will die in their 30s, if not earlier. But today, with improved medical interventions, people are living a fairly full life with cystic fibrosis. Unless they encounter something like Pseudomonas aeruginosa, which is their worst nightmare, because this bacteria is just, it grows very well in that mucus environment. And it further paralyzes the cilia on those respiratory epithelial cells, so it makes it very difficult for them to clear those infections if they get it. Add on top of that the fact that anytime you have a patient who has an opportunistic Pseudomonas aeruginosa infection, it's just super difficult to treat with antibiotics. So it becomes a very life-threatening situation if those patients have a Pseudomonas aeruginosa infection. There are some GI infections. You can get mild diarrhea. And some patients who are predisposed, like infants and people who are like cancer patients, for example, could get necrotizing enterocolitis. But probably the main things to associate with are wounds, burns, and respiratory infections. For systemic infections, you can get bloodstream infections, bacteremia that progresses to bloodstream infections, secondary pneumonia, osteomyelitis, joint infections, particularly with IV drug users, more so than with UTIs, with that as well. Again, with IV drug users, endocarditis is a possibility. In the tricuspid valve especially, this is where those bacteria tend to start seeding. And then we have some CNS infections as well, brain abscesses and meningitis. These are possible, but they're not nearly as common, so they're fairly rare with Pseudomonas aeruginosa. But still, that's an impressive array of things, localized and systemic infections from one bacteria. And these are going to be hospital-acquired primarily in hospital settings because it's so difficult to eradicate. In terms of antibiotics, this is one of those escape pathogens. The CDC has ranked it as serious. WHO ranks it as like urgent level priority for developing new treatments against it because of the amount of resistance that it has. When we talk about this bacteria, this gram-negative rod, it has in its outer membrane in lower permeability, so a lot of antibiotics simply cannot get in. That's one of its intrinsic methods of having antibiotic resistance. It also has a lot of efflux pumps, so it's hard to get targeted concentrations of antibiotics within those bacterial cells because they just keep pumping them out. And then it's very good at acquiring resistance from other bacteria. So this is how we get, when we start drilling down on some of those isolates that turn out to be MDR, XDR, or PDR strains, pan-drug resistant strains, some of those antibiotic resistance genes are being transferred from other bacteria, so it seems to be adept at accepting those. Then when we talk about adaptive or inducible resistance, the biofilm production is a significant source of that resistance, up to 1,000 times more resistant to antibiotics than bacteria that are planktonic and individual off on their own. And those are, like that biofilm formation, might have mentioned this earlier, but that's triggered by a critical mass of bacteria that develop quorum sensing. So as soon as a good aggregate number of them assemble, they'll start releasing small molecules and start upregulating alginate expression to contribute to biofilm formation. All right, any questions about Pseudomonas? Okay. Now we'll move on to another biological threat agent. The first one we talked about was Staph aureus enterotoxin B, which wasn't the organism itself, but it was the toxin produced by Staph aureus. But this agent itself is considered a biological weapon. It's extremely rare. If it weren't for the fact that it can be weaponized and has been weaponized historically, we wouldn't even be talking about it. It's an example of a zoonotic disease. It causes something called glanders in horses, if you've heard of that. And even the human form, when it affects the lungs, is also called glanders. But what it does in horses is just causes this massive pneumonia, this respiratory infection in horses that can be fatal for them. You can see like the snot kind of dripping out of this horse there. So they are social creatures. They transmit it pretty easily between them. And that material can be infectious for humans as well. So if you come into contact with respiratory secretions from infected horses, you can pick it up yourself. That puts occupational risk up there for disease acquisition. If you are working with horses, veterinarian, whatever, you would be at a potentially higher risk for acquiring it. It is possible to spread from person to person, but that's not a significant source of this disease. It would be relatively difficult that way. This is how bad it was. This is an historical photo from back in, I don't know if it's dated there, but I don't see a date, maybe around World War I, when they were still using horses in that generation for war. It was definitely used and weaponized in World War I to target horses because they were used in supply trains. But they had to keep horses separated so they wouldn't use the same water bin together. They had a bunch of other mitigations that they used to prevent transmission there. So, in terms of transmission, this is categorized as Tier 1 select agent by the CDC, which means it has a high risk of being used for malign purposes. It can be aerosolized, which means you can, if you're able to aerosolize it, you can distribute it to a wide group of people and infect a lot of people. And it also has a fairly low LD50, so it makes it a more virulent pathogen. In terms of the disease, I've mentioned that glanders affects the respiratory system. Farcy is another manifestation that affects the lymphatics and the skin. You can have both. This is a death mask taken from a veterinary student who died of Farcy. I think this is late 1800s. You can see the skin nodules. These are called Farcy buttons that appeared all over his face. And they were good enough to take a death mask to remember what the pathology looked like, I guess. There has not been a case of glanders in the U.S. since 2000, so I put the link in here for that case report that was written up on the last case we had, if you're interested. It's a very interesting case. It happened at USAMRIID, and the guy was working with Burkholderia malleii. He was immunosuppressed, he was diabetic, so he had a comorbidity, and he got very, very sick. And when he got sick, that's what the write-up is about, how difficult it was for that presentation to be linked to this etiology because of how rare it is. They put him on at least 4 or 5 different antibiotics in that case report because they couldn't figure out what the etiology was. His symptoms were mystifying. They weren't able, he didn't have this Farcy presentation, so it was mostly like a pulmonary disease, and then he also had some organ involvement as well. But they finally diagnosed him because they had some imaging, and it looked like there might have been an abscess in his liver, so they did an FNA, fine needle aspirate, of his liver, and then they cultured that. Very slow-growing organism. They finally grew up the organism itself and then misidentified it as Pseudomonas instead of Burkholderia, which is understandable because they do have this very similar composition. But it was difficult to get that confirmatory diagnosis and put him on the right antibiotics and save his life. And so that's an interesting case report. It just illustrates part of the problem with that. And another reason why it's such a good biological weapon, because it's hard to diagnose. It'd be very tricky to diagnose that. Very, very fatal, 95% fatality rate if you don't treat it. And if you do, it's still very, very, very high fatality rate. Also in this genus is another biological weapon, Burkholderia pseudomallei. This has some similarities and some major differences from mallei. It's kind of hard to keep them apart since their names are similar enough. But this one isn't so much of a zoonotic, so it's not like a disease that's zoonotic in horses, but it is an environmental bacteria. So you acquire it, usually the main source of transmission is acquiring it from inhaling contaminated soil. That's classically how you get it. Historically, in the US, this kind of hit our radar during the Vietnam War. We had a bunch of cases of melioidosis, which is a disease that's caused by Burkholderia pseudomallei. In Vietnam, vets that were coming back from deployment, and they would, after months, sometimes years, present with this very difficult to diagnose disease, and they eventually traced it back to, or identified the cause of aging. But how they think they got it oftentimes is from rotor wash, from helicopters that were coming in and picking up those soldiers. So that's kind of like the classic association with Burkholderia pseudomallei. But as a biological weapon, many of the same reasons apply: aerosolization, low LD50, multiple routes of infection, high mortality rates, difficult to diagnose, all of that stuff. Intrinsic drug resistance, hardy bacteria. Vietnamese time bomb, that just refers to how long it takes to incubate sometimes. That range can be anywhere from days to months to years. And when you grow it up, it does take a long time, but it has these very pretty violet-colored colonies that have a wrinkly morphology to them. In the US, I'm not aware of many cases, but there is a link down here to an outbreak that happened during the COVID years in 2021 recently. It was linked to some aromatherapy products that were made by Better Homes and Gardens and distributed by Walmart. Yeah, this is like in I think that's chamomile and lavender mist thing or whatever that was sold. But the product itself had been manufactured in India. They contacted the manufacturer after there was a small cluster of cases and tried to see if they could nail down which of the ingredients was contaminated with this bacteria. But there were at least 4 people who got this rare exotic disease. There was one in Texas, one in Kansas, I forget where the other 2 were. And all of these people were also sick, so they weren't healthy people. This was during COVID. I think 2 or 3 of them had COVID, and that might have been responsible for that death. But really strange outbreak. Bizarre. Would not have wanted to have to diagnose that one. In terms of the disease presentation, very difficult, like I said. Its nickname is the Great Mimicker because it presents like any number of things, especially tuberculosis, which we'll talk about early in Block 4, but it can affect virtually any organ system. It depends heavily on the route of acquisition. So if you're inhaling it, you will probably get something like a pulmonary infection, a lung infection, pneumonia, or if you get it contacted to an open wound, skin infection, ulcers, nodules, abscesses, things like that. A rare picture of a guy who has melioidosis. You can see this very swollen lymph node in his neck. That's just the way he presented. I don't know what his route of exposure was for that presentation. This looks like a liver. It has been it's on an autopsy, but I think it's from an animal. I think both of these images are from animals that were experimentally infected with Burkholderia pseudomallei. And it's just caused massive necrosis in the liver, and these nodules have started to form in some of these viscera, the internal organs. This isn't a goat that was infected. So just a wide range of symptoms. The organism itself, might not be shed in huge numbers, but it's shed in a lot of body fluids. So if somebody does have melioidosis, you would want to make sure that they were probably quarantined, or at least these specimens were treated with care, so you don't get lab-acquired infections or other healthcare, like transmission to the health provider themselves. With Bordetella, you may have heard of this one before. Bordetella pertussis. Did you have a question, Aaron, on that?
No, it's just my face.
Oh, okay. Fair. I mean, not a dig. Bordetella pertussis. In this genus, Pertussis is not the only organism that can cause respiratory infections, but it is, again, is the most important one. When I was at Madigan, we had a bunch of Bordetella cases. So we had a PCR that we developed that was a nested PCR, and we had primers against other species like Bronchoseptica, Holmesii, Parapertussis, and pertussis itself. We were catching all sorts of cases of Bordetella caused by those other organisms. But that said, like respiratory infections, all of them kind of contribute to respiratory infections. That said, this one is definitely the most clinically relevant one. Historically, there have been big outbreaks of whooping cough before we had the vaccine widely available. I don't know how many people even appreciate how devastating this disease was before the vaccine, but certainly a lot of infections, and particularly bad with kids. It's usually considered a pediatric disease because they have smaller airways and relatively immature immune systems. They tend to get, if there were any fatalities, it tended to be in the pediatric population. So if you think it's gone away, it hasn't, even though it's in that Tdap or DTaP vaccine. We still had about 19,000 cases in 2017. Fortunately, most of these have been on decline, except for I do have some data in a couple slides to show you about the slow, steady increase in cases in post-COVID years. It seems to be, it's a little bit cyclical, but we're also worried that there might be some vaccine hesitancy with Tdap and DTaP. What it actually causes is whooping cough, more commonly known as whooping cough. This is a really serious infection where you have extremely violent coughing. Quick note before I move on to the next slide, strictly human disease, that's with pertussis. Bronchoseptica, for example, causes similar symptoms, not as severe, but anybody heard of Bordetella bronchoseptica before? You have? A couple people maybe? Kennel cough, yeah, exactly. That's dogs are mostly affected by that. It is potentially transmissible to humans, but when you board a dog, they pick up kennel cough. Bordetella bronchoseptica can also be that could be considered a zoonotic disease that can be transmitted to humans. But this one, strict human disease. All right. In terms of what happens, this is acquired by inhalation. You inhale bacteria, person to person. And those bacteria will get into your respiratory airways, and they will adhere to respiratory epithelial cells. This is a three-stage disease when you're in an exposed, susceptible person. It starts with this heteral stage. This can last a week or two generally. Incubation periods can go almost all the way up to 3 weeks sometimes, but generally less than 2 weeks. And in this heteral stage, what is remarkable is just the proliferation of bacteria in the lungs. That's primarily what's happening during that time period. So the symptoms are mostly cold-like symptoms, nothing typically or terribly distinctive. Once you get into that second stage, this is when it becomes apparent that it's whooping cough. And at that point, it's mostly toxin mediated. So it's the pertussis toxin that's responsible for a lot of the really serious violent coughing that characterizes this disease at this point, stage 2, proximal stage. The classic whooping sound is very distinctive. It's like this gasping breath. I have play you a little clip if you've never heard it before. So, hear that. This is a baby with whooping cough. Can you hear that? So, that baby is struggling to get air, and some of these coughing fits, these pertussis fits, can be so violent that kids, especially kids, can become cyanotic, they can become blue because it's so hard to get oxygen into their lungs. They can have subconjunctival hemorrhaging, so their eyes, like the blood vessels in their eyes will pop a little bit and they'll get bleeding there. Just like related to pressure, the tongue can start protruding, the eyes can bulge, you can get petechia on the face from burst blood vessels on the face. And then after these fits, which can last like 15, 20 minutes, and you can imagine trying to sustain that for 15 or 20 minutes. After that, a lot of people will vomit or they just are completely exhausted. I mean, they are wiped out and fatigued from that. Not to mention the anxiety they have because they know that another fit is coming on. It's a very, very distinctive disease. If they it's not a huge mortality rate, but if they do survive past the paroxysmal stage, convalescent stage, you'll see a tapering off of the whooping cough symptoms, but this is when secondary complications are oftentimes associated because the lungs have been beat up so much and those tissues have been really degraded over that experience for a couple weeks. This can last up to 8 weeks, so very long time. Treatment is mostly supportive, but there are some antibiotics that can be administered as well. Why would treatment during the paroxysmal stage be mostly supportive instead of antibiotic treatment? Because it's toxin mediated. So if you're going to prescribe antibiotics, it would probably need to be in the catarrhal stage when the bacteria are proliferating and reproducing for it to be most effective. And that's when it would be most difficult to recognize and diagnose as Bordetella pertussis. In terms of virulence, as I said, primarily toxin-mediated disease, they do need to attach, and these attachment factors are important for the vaccine. We'll talk about that in a little bit, but they've got protactins, hemagglutinin, which causes sticking to red blood cells, as the name suggests, and then they have Fimbria to allow them to attach to those epithelial cells. For localized damage, they have this dermonecrotic toxin, causes localized ischemia. What is ischemia? Lack of blood flow to an area, so the tissue is deprived of blood flow. Tracheal cytotoxin, this will paralyze cilia, so they're not functioning properly. This is essentially, that tracheal cytotoxin is basically just a chunk of peptidoglycan, a specialized monomer of peptidoglycan with a disaccharide in the pentapeptide chain that gets released from the bacteria, and it causes that kind of effect on those epithelial cells. And then lastly, you have the pertussin toxin itself, and that's what's responsible for systemic toxicity. So that pertussis toxin is also cytocidal. It will destroy those cells and reduce their ability to contribute to respiratory secretions and mucus production. For the vaccine, I mentioned DTaP and Tdap. These two different vaccines are basically different amounts of the pertussis toxin. So this is diphtheria, tetanus, and acellular pertussis. It's the protactin that's the acellular component. The first vaccine against pertussis was introduced, you can see this on the bottom there, back in the 1940s. So that was a whole cell. You had the inactivated whole entire cell of pertussis in there, Bordetella pertussis. And that worked well. You can see the number of cases, the incidence per 100,000 dropped hugely, just plummeted. The problem with that vaccine, though, was that, and it was also diphtheria and tetanus, it was a trivalent vaccine. The problem with the whole cell vaccine is that it caused a lot of side effects. So great immune response, but a lot of side effects. So in 1990, they reformulated it to make it better for kids, and they introduced DTaP. DTaP had very few side effects, but it didn't offer the same level of protection. So we do see an increase in cases, but we consider that to be acceptable because the side effects are so severe. And then in the mid-2000s, we introduced Tdap. That's the one that is formulated where it actually has a little bit, I think, less protactin in that vaccine. And that's the one that we use for the adult population, adolescents on up. It does need to be boosted. And for pregnant women, that's it's allowed for pregnant women. In fact, they recommend getting boosted with every pregnancy with this vaccine, so it's safe for that. The strategy there is you want to have some sort of cocooning effect for the baby when it's born, because you can't vaccinate before 2 months of age. So you want to vaccinate all the members of the household so the baby won't pick it up from them. Sorry, it's 2:08. I'm sorry, guys. If you guys want to take a quick break, there's enough to go for a little bit longer. 10-minute break. Okay, I'm so sorry. 2:18.
I did. I highlighted like something like that. But there's a lot of repeats that
Isn't it like itself? Like I'm over dominant. It was literally a dominant like walking through the house. My wife was This is just pretty good at
The craft is good, they have to trust us.
Like I wasn't a big fan of raw sushi for a while, and then like when I started eating more than cooked ones, and I started getting more into it, and I was like, oh, I can do this. You know what's funny? I get sick from sushi, like 3 out of 4 times. It's not even sushi. It's like thick rice. Because like the rice and the rice and so it's prepared so that it tastes so like so basically like I honestly don't know what I do, but like that's because like you have parasites right? And honestly, I think it's like my theory, like it has to be non-wild non-wild. So like it's like this year, for everyone like get like all the way to so it's better and they put points like something like
Why don't you like what
Oh yeah.
they have like the big crab.
but it's like raw sushi. But like, I'll take crab.
You don't want to eat no.
Yeah, no, I think I think crab.
Extra fish sauce. Yeah, that's fine.
Yeah.
Broski's pretty good. Broski more Polish.
Yeah. I think it's all like the biggest risk you take is like because like only like certain chefs can make that scale of content guy. So I know with like fish, right? you ever see like a big like cinema poster in Japan? They have like giant. But it's like, think about it. Anything like a grocery store, you're relying on someone to write it. Always farmers, the processors, the person actually putting it on the that doesn't mean like that meat has not been like bought. It has to be strictly off.
Well, yeah.
So it's like, unless you're here to sell, it's like a big thing about anything, right? Like you driving here from your house, you have to approach the person out the lane, do the right thing, stopping at a red light. I think I gotta hope whoever made the building knows something about architecture that should not have a law. What else is it? We're going 70s, so 50 years later. That's how it goes.
High pride.
We had a 3 o'clock class. Your boy would be.
Friday, finish here, and you move the rest of it like that one day. You're gonna be here, and it's gonna have to bad. You're gonna have to continue on that. Oh, really? It's so funny. Like, people always ask me for that I would have to do. Like, nothing to one better. It's like, how dare you? It's like,
Goal was that way. They walked the whole way. That's the way I think about I'd admit it's 3 days.
Yeah, like you can't hear them because there's so like there's some stuff smoking.
Yep.
That's also good.
Which is crazy because the recording.
So it's like you watch the recording, you hope people hear it, which I can watch. Oh, I can barely hear it.
No.
Yeah, like I could barely hear it.
Did you wear your earphones?
I'm putting it on. Like, I'm also deaf, so it might just.
I thought I could hear him.
Yeah, it's kind of hard. I don't know what it was. It was the fourth one. I'm sure I was the second.
That's what you take?
Who knows? You always need us.
Okay, so we have 7 slides left to go, and then we're free for the weekend. We just need to talk about a couple more organisms, starting with Legionella pneumophila. Pneumophila tells you what about this organism? Lung-loving, yeah. But a little bit of a misnomer. It is another one of those environmental bugs. It's just doing its thing out in nature, living in aquatic systems. But what makes it adaptive for humans and able to cause infection is actually the fact that it is adapted to live inside of amoeba out in the environment. That's normally what they do. So when they get into the human host, the reason why they're able to establish infection is because they are able to replicate inside a cell. So you can call them facultative intracellular parasites, if you will. Remember in block 2 when we were talking about organisms that weren't really on our radar until we had changing social behaviors and things? We mentioned Legionella pneumophila there. Remember what it was associated with, where it became suddenly on our radar? HVAC systems. How timely, right? So this has been around for a while and has probably been causing human infections for a while, but it came onto the scene in a very big way, July 4th, 1976, in Philadelphia. Anybody familiar with that outbreak? It was a gathering at this convention center of over 200 veterans who were members of the American Legionnaires, American Legion. They were Legionnaires, members of the American Legion, and they were celebrating the bicentennial, getting together every year. So these were veterans. This is in 1976, so probably a fair number of young guys coming back from Vietnam, guys who were a decade older, coming back, 2 decades older, coming back from the Korean War, and then add on another decade, you had a bunch of World War II vets. So these 200 attendees tended to be pretty old on the age distribution scale. A lot of them had comorbidities. A lot of them were smokers. This is in the 1970s. A lot of them drank a bit, maybe. So a pretty vulnerable population together in one building in the middle of the summer where it was hot and AC was starting to be used a lot. Out of those 200 plus people, 81 got sick mysteriously with this very aggressive respiratory infection, this pneumonia that they developed, and 31 of them died. As a nation, we were panicked because nobody could figure out what the cause was. Epidemiologists were doing a lot of work trying to trace what the source was. They could not establish what the source was. And up to that point, they had never identified and characterized Legionella pneumophila, which they eventually finally did. So, and after that, we had big impacts. We modified building codes. We modified how water handling systems work. We put a lot of money into research about Legionella pneumophila. So a lot of this is sort of like stems from that big outbreak, but we've had several outbreaks since then, and we get them every year. We get clusters of disease caused by Legionella pneumophila. As I said, out in the environment, this is a water bacteria. It gets into our buildings, into hot tubs, cooling towers, certain medical devices, decorative fountains, misters. A place like Texas, we have misters all over the place, right? That's a perfect source if that water is contaminated for spreading and aerosolizing Legionella pneumophila. It's pretty, like most environmental bacteria are, it's tolerant to a wide range of different temperatures, so that contributes to that as well. And because of the way this infection happens, which we'll talk about in another slide, when you get these infections, they tend to, you cannot transmit them very easily person to person. So the main transmission source is just inhaling contaminated water, not from an infected person transmitting to the next person. They just keep that in their own lungs, primarily because these are intercellular and deep in the lungs. All these factors lead to Legionella growth. I don't think we need to go over all of them. They do form biofilms, and that's what this cartoon in the upper right is showing. That you can get a layer of Legionella bacterial cells that can grow. It's very difficult to eradicate those. But yeah, just something to keep in mind there. Here we are, the increase in the number of cases that have been reported. Part of this is not just the increase in the use of air conditioning, but part of this is because we've gotten better at identifying it. Our providers, our doctors know what to look for. It's in their differential diagnosis. We know how to grow it up in the lab better now. It's not the easiest thing to grow, but we can do a pretty good job now. We have PCRs for it. So this big uptick is it's attributable to a lot of different things. In terms of the diseases it causes, there are two diseases. One I was talking about was the more serious one, Legionnaire's disease. Legionnaire's disease is characterized by a very rapid, acute onset of fever, non-productive cough, headache, chills, but the important thing is that they have pneumonia, severe pneumonia. So it tends to happen in people who have some sort of underlying comorbidities. With Pontiac fever, you can get the same symptoms but minus pneumonia. Patients who get Pontiac fever tend to be immunocompetent, and even these guys, even if they're healthy, there's probably a dose factor in here too. So healthy people, if you give them a big enough dose, they might develop Legionnaire's disease. But more often than not, the cases that progress to actual Legionnaire's disease happen to people who have these serious comorbidities, chronic lung disease, COPD, chronic obstructive pulmonary disease, things like that. Even immunosuppressed people, people with cancer. So, and with it being facultative intracellular, this microscopy image that we're looking at, you can see a couple of those bacterial cells, these dark bacteria. Some of them are outside of the cell and some of them are inside the cell. So they can survive in either environment, but they replicate in those cells. This you definitely don't have to know, but Bob Dylan did write a song about it in 1981 about Legionnaire's disease because there was so much confusion caused by that outbreak. It took years for them to pin it down, and you can kind of see by the lyrics. This is a terrible song, by the way, if you ever listen to it. Not a good song. I have a whole playlist with infectious diseases, and and I have anthrax on there and contagion, which I don't like, but yeah. Anyway, this is not one of my favorite ones. But the lyrics are really good, right? He's the bard, he writes fantastic lyrics, and it captured the national fear and sadness that happened after that outbreak, and the inability to figure out what was causing it. Intracellular pathogen, how is it able to survive, primarily in alveolar macrophages? I should have put the word alveolar in front of that, but also in the parenchyma of the lung as well, so the alveolar epithelial cells and the macrophages. The primary way that it's able to survive once it gets engulfed and phagocytized by these macrophages is by escaping this phagosome and making its own little vesicle that it can replicate in those cells. So it can replicate, and in the process, as it's starting to reproduce, it manages to escape immune detection, as most intracellular pathogens do. When you do mount an immune response to it, it's mostly T-cell mediated because, like any T-cell response, what's being recognized by the cell is being presented on the surface of an MHC class 2, right? So it's not, these are not extracellular bacteria. Some of them will tip off a humoral response, but most of them are going to be triggering the T-cell response. Those that do actually end up getting processed and presented as antigens on the surface. In terms of virulence, they will stimulate a lot of cytokines to be released. The sensors in these cells, like the NOD system, we haven't really talked about much, but the NOD system will go off and you'll get a lot of cytokines that are produced. So that will contribute to immunopathology, inflammation, some of those respiratory symptoms. They have LPS and pili. And then again, they just prevent the formation of that fake lysosome to escape into their own little vesicles. For lab diagnosis, we have a particular kind of media that we can use to culture sputums and other respiratory samples, like deep respiratory samples. The bronchoalveolar lavage is the preferred specimen because it'll go deep into the lungs. This is a charcoal-based media type, and it is selected for Legionella pneumophila. These might take some time to culture, but this is still the gold standard. It's still something you'd want to do for every specimen where you suspect Pontiac fever or Legionnaire's disease. Also, in conjunction with that, we have an immunoassay. This is a rapid antigen test that's made by Binax now for Legionella. We also have ELISAs that we can do as well. Strangely enough, we can do these. They're sensitive enough to pick up antigens from this respiratory pathogen that are released in urine. I don't have a good grasp on how those antigens get into our urine, but probably they get taken into our bloodstream and then they're cleared in the kidneys or something like that. But this is it's considered to be a pretty sensitive and specific test. So we do it in conjunction with the culture of the BEL, and we base our diagnosis on both of those methods. Any questions about Legionnaire's disease or Legionella pneumophila? Okay, Moraxella catarrhalis. This is mostly a respiratory pathogen. Intracellular gram-negative cocobacilli, or cocci. So you can see very clearly here a bunch of those in most of those bacteria inside a cell. It is normal flora of the upper respiratory tract. It's a little bit misleading though because it's not a dominant species in the respiratory tract, and as we age, we get less and less of it. So the people who have the most Moraxella colonization are usually the pediatric population. As a result, a lot of the infections from Moraxella tend to be in kids. So we might be colonized to a certain extent, but just not as much as kids are. It also does very well under oxygenated conditions, but I think it's not a strict obligate arrow. It might be somewhat facultative. Types of infections that it causes are going to be really important for you to remember, especially next semester, but significant contributor to otitis media, conjunctivitis, these eye infections, and sinusitis. So mostly, again, these upper respiratory tract infections. If you have people with comorbidities or older patients, bronchitis or pneumonia are possible. And very, very rare, sometimes you get joint infections and meningitis. This is not a major cause of either of those infections, but it is possible in immunosuppressed people. In terms of virulence factors, nothing that we have not seen before, but that beta-lactamase production is notable for treatment. And because of that, you want to do, certainly want to do AST testing if you're going to use antibiotics. Acinetobacter baumannii. This is the last organism we're talking about today. Another Cocobacillus. It stains a little strangely. This is a gram-negative, but on this gram stain, it's the organism on the right that almost looks like a gram positive. It's staining very purpley. It's just the way it takes up stain. But you can see how that might be confusing. Next to it, we have some Pseudomonas, which is a nice, like, proper gram-negative stain, and it's also the morphology is much more classic. So gram-negative rod, Pseudomonas looks like it should, but Acinetobacter baumannii, which is also gram-negative, looks almost gram-positive, and it is coccal bacilli. So very stubby morphology there. Kind of hard to look at, or confusing to look at. When you grow it up, it has that nice pastel pink colony appearance, very characteristic for Acinetobacter. The name baumannii is named after the person who described it, probably, but Acinetobacter is derived from the Greek from akineto, no movement. So these are non-mobile bacteria. Also associated with opportunistic infections and hospital-acquired infections, hospital environments, mechanical ventilation equipment, so pneumonia cases, VAP, again, ventilator-associated pneumonia. And this statement about them being part of the normal oropharyngeal flora, that too, it's true, but very transient. They don't live there long. Also worth noting, this is a remarkable feature about Acinetobacter, the fact that they can survive on dry surfaces for long periods of time. We're talking about hospitals that have Acinetobacter kind of just ubiquitous in the environment. It's because they can survive with very little liquid for very long periods. It makes it hard to disinfect. The range of infections are those you kind of expect with other hospital-acquired infections that we've talked about. Pneumonia, bloodstream infections, UTIs, wound surgical site infections, meningitis, septicemia, all of those things are associated with people who are coming in for treatment and getting exposed to the bacteria in the hospital. Where it came on the scene for us, particularly with the military, was early on in the 2000s, during the height of OEF and OIF. We had clusters and outbreaks of Acinetobacter from some of the combat support hospitals and some of the ships, treatment ships, where patients were coming in for injuries and they were getting these really difficult to treat infections from Acinetobacter. We, for a long time, did not know whether they were getting those infections because they had like a penetrating injury from shrapnel or some other penetrating projectile that had the bacteria contaminated on it, or if they were getting the infection after the trauma once they were in the hospital. So we finally figured out that transmission was mostly because they were getting it in the hospital, not from the penetrating injury itself. Very, very famous in its association with those wars. It has the nickname Iraq-Bacter for that reason. But that's not to say that in normal clinical areas, civilian hospitals, this isn't a big problem. It absolutely is. So any normal hospital patients can pick up Acinetobacter. This study, it is the A in escape, so you know it's going to be a big antibiotic-resistant bacteria, right? This study I'm including, it's super dated, it's 21 years old, but I put it in here for historical interest more than anything. If you want to read through it, it's not testable, but it has some good information about early on findings about the level of resistance, the fact that they found, they were characterizing these isolates, or these clusters, 4% of those isolates were pan-drug resistant. Now we think even higher percentage might be pan-drug resistant. So it's just a very scary drug for that reason, since it's especially resistant for carbapenems. It has lost its susceptibility to a lot of cephalosporins as well. So, and even colistin is sort of one of those last line drugs, and I think there might be some colistin resistance as well, which is very, very difficult to treat. That's all I've got for you guys. Have a great weekend. Let me know if you have any questions, okay? Thanks, guys.
Did you get that?